Welcome back to the School of Muscle podcast. Today, Dr. Roberts joins me to discuss a study that he was an author on, and this study was titled Molecular Predictors of Resistance Training Outcomes in Young Untrained Female Adults. And that is what we are going to review today, and I'll make sure to have that study linked in the description and all the details in there as well if people want to go kind of check that out. So I think a first kind of good place for us to start here, Dr. Roberts, is what kind of led you to wanting to do this study in the first place here?
Yeah, right. That's a good question. So my lab, we sort of deal with skeletal muscle, and a main thrust for our laboratory is examining mechanisms that either coincide with or are involved with skeletal muscle hypertrophy when you weight train. And a large part of the literature that still needs to be explored are biomarkers, or I would say molecular markers in skeletal muscle that respond to training in like people that gain a lot of muscle and may not respond so much in people that don't gain a lot of muscle with resistance training. So the field has termed this as responder, non-responder analysis. And there's been a lot of data in young untrained men. And so we had an opportunity, we had a data set sort of in hand. We had muscle from women that trained with us for 10 weeks. And we said this would be the perfect opportunity with this many females worth of tissue that we had banked to examine some of these molecular markers. And so that was certainly the motivation of the study. What got me into the topic in general was a researcher by the name of Marcus Baumann. So when I was coming up as a master's student and a PhD student, I read Dr. Baumann's work. He and I to date are close friends because I'm at Auburn University here in Auburn, Alabama. And I would say until about 2 years ago, Marcus was at UAB in Birmingham at their medical center. And so we would cross-pollinate a good bit. We got to publish together, which is great because it's like, you know, imagine having a hero and then getting to work hand in hand with your hero. But Marcus had some really nice work in the mid-2000s where he sort of sparked interest in this research topic. So when people resistance train for, call it 10 or 12 or 16 weeks, you know, what are some of the molecular markers and muscle that delineate higher versus lower responders, those that gained much more muscle mass? In the case of Marcus's work, it was muscle fibers themselves, right? Extreme responders showed this very robust increase in size with resistance training, and non-responders showed no increase in muscle size. So I would say Marcus Baumann got me into that. And then since then, Stu Phillips at McMaster University, he's engaged and he's done a couple of nice papers on the topic. Beyond the paper we're going to talk about today, we've done a few other papers as well on the topic in terms of looking at biomarkers that may delineate higher versus lower responders in different populations.
Perfect. And towards kind of the end of reviewing this study here, we'll definitely maybe cover whether this agrees or disagrees with other research kind of in this area, and we can kind of go over that as well. So I think that sets things up really nicely here. It's definitely a topic I'm super interested in, and I know that generally for people like listening to a podcast about the science of like growing muscle, I feel like sometimes those are the exact people that maybe are lower responders and have a harder time kind of building muscle. So I'm sure that they're quite interested in this as well. So I think with that setup here, what were kind of the methods of this study? What you guys kind of decide to do here to try to tease out some of these molecular markers of kind of higher and lower responders to training?
Sure. Yeah. So we had around 34 female participants. They all partook in baseline testing, which involved them coming into the laboratory, getting body composition assessment with what's called a DEXA device. And so that's very, very low dose radiation. It does a full body scan. You would see that in a clinic for osteoporosis, assessing bone mineral density. It also can tell you whole body lean tissue mass, and it can tell you whole body fat mass. And so over time, if you were to do like a pre-intervention DEXA and then go resistance train and do a post-intervention DEXA, if you were to see an increase in whole body lean tissue mass, that would be more or less indicative of gaining muscle mass, because the presumption is you're not going to see any of the visceral organs appreciably change in mass, like the liver, intestines, and so on. So baseline testing, these 34 females, they came in, they had a whole body DEXA scan. We got some strength assessments. So we looked at bench press strength, we looked at leg press strength, we looked at deadlift strength. We also did some thigh musculature scans that were specific to the upper mid-thigh of the right leg. And then we took a muscle biopsy out of the upper mid-thigh, sort of the outer portion from the vastus lateralis. So baseline testing, to recap, right, was sort of whole body composition, mid-thigh muscular area assessments, and then a small biopsy from the mid-thigh. All of these 34 participants then engaged in 10 weeks of resistance training. Training was 2 days per week. There was 1 day per week that was kind of a higher volume day, less intensity, if you will. The second day per week of training was a little bit lower volume, but higher intensity. And then in terms of the workout per each of these 20 sessions, it involved 5 exercises. So we had a leg press with like a hip sled, right? We had a barbell bench press, seated leg extensions, hex bar deadlifts, and then cable lat pull downs. Beyond that, so this was a secondary analysis, gets a little bit into detail. I would say half of these participants were consuming a peanut protein supplement, the other half were not. However, that supplement had no effect on any of the outcomes we were looking at, so pooled everybody into this responder analysis, all 34 of these females.
Gotcha. And did they have any prior experience with training, or were they all kind of novices when it came to training, or what did their kind of experience look like?
Yeah, I mean, so it's kind of a crapshoot for those that aren't involved in recruiting and testing, human participants. You're going to get all walks of life. So the demographic was college age. On average, these 34 females were 21 years old. I would say some had some experience in high school with athletics, but I would call most of them having not resistance trained for 2 to 3 years. When you look at their strength metrics and you compare it, for instance, to trained females, their strength metrics were appreciably lower. So we would say that maybe some had some prior training experience, but by and large, they were certainly novice trainees.
Gotcha. And then you mentioned the protein there. Were there any other differences regarding their nutrition? Like, maybe someone listening to this might think, well, if one group just was eating sufficient protein and the other group wasn't, maybe they would see better results. But did you see any differences there when it came to nutrition?
We developed sort of this multifaceted analysis to say, okay, this person was a higher responder with pretty good confidence. This person was a lower responder with pretty good confidence. So I'll just get into the way that we did that, right? In all 34 participants, when you look at pre versus post intervention, this is training for 10 weeks, 34 participants worth of data, we saw a 2.5 pound increase in lean tissue mass. We saw a 50 pound increase in deadlift strength on average, and we saw around a 15 pound increase in bench press strength. Now, again, that's in all 34 participants. When we did the clustering for higher versus lower responders, and we look at these things, this is where you can appreciate, right, there was certainly a divergence. So the higher responders gained 5 pounds of lean tissue mass or muscle. The lower responders on average didn't gain any muscle. That's difference number one, right? Second, looking at the mid-thigh musculature, the higher responders showed around a 10-time increase in thigh muscle gains than the lower responders. And then finally, the higher responders gained 80 pounds on average for their deadlift strength, and the lower responders gained 30 pounds. So we like this multifaceted approach. When these studies were first being done, researchers would hang their hat on one variable, right? And that is to say that, I may differentiate high versus low responders based on, the changes in muscle fiber size. The problem with that is that every technique has error. And so to more confidently say that someone's a higher versus a lower responder, we feel like you need a lot of different techniques to confirm that observation, right? Hence, our philosophy is take the whole body musculature response and quantify that pre to post intervention to say, okay, well, these higher responders gained 5 pounds on average, lower responders didn't. Check. The next thing is to go more localized and look at mid-thigh muscle imaging. And again, we confirm that in these same higher versus lower responders. And then finally, we wanted to bring a functional strength outcome to the mix, right? And so that's why we chose to allocate deadlift. So we like the multifaceted approach because some scientists would certainly criticize like, you shouldn't be doing high versus low responders. Some of these results could be regression to the mean, which we won't get into too much here. And so we said, fair point, but clearly we've done enough training studies where we look at a participant and we're like, wow, you've gained a lot of muscle. You are a really good responder, right? So we think there certainly is something to it, but you just need to bring this level of rigor and more confidently calling somebody a higher responder and somebody a lower responder.
Yeah, and I personally really like that. As in practice as a coach the last several years, it's sometimes hard to determine if a client is making progress, especially if you just look at one specific variable.
Yep.
And you can increase your certainty or increase the probability of progress if you look at, strength outcomes, photos over time, seeing visual changes and measurements, different things like that. So I really like that you guys use kind of a multi-factor approach there. So clearly with your study, you definitely saw some major differences between individuals in which, a lot of people might read the abstract of the study and see individuals gain, 15 pounds on average. But when you see that huge difference with some gaining like 30 and some gaining 0, it makes you think, okay, there's definitely something going on here with higher and low responders, which might also lead people to kind of want to consider what's kind of going on here at a more mechanistic level. So did you guys see certain kind of molecular differences between kind of the higher and lower responders, or what were kind of the results when it kind of came to that side of the things?
Yeah, for sure. So we did, right? We really had a priori, so prior to even getting into this tissue, there were 3 molecular targets that we were going after. So one would be the muscle protein synthesis response to the very first bout of training, right? We hypothesized that higher responders after lifting weights for the first time, session number 1, would show a higher muscle protein synthetic response compared to lower responders. So the study design gets a little bit more complicated, and for those interested in that approach, they can read the paper, but that involves giving people stable isotope tracers, safe to consume, no problems there. It's been done extensively, but we had to do that with tracers and biopsies. So hypothesis 1, muscle protein synthesis responses, because as I'm sure you're aware, Ryan, and some of your viewers, there's a lot of literature on the importance of muscle protein synthesis for building muscle, right? All right, hypothesis number 2 was that we would see better increases in muscle ribosome content in higher versus lower responders. So without getting into too many details, the molecules in the muscle that catalyze muscle protein synthesis are called ribosomes. And when you lift weights, you will see an increase in a process that's called ribosome biogenesis. And that is the muscle cells creating more ribosomes as the cell is growing with resistance training. There's a lot of literature to suggest that with muscle hypertrophy, ribosome biogenesis is required, right? And there's even been, we've put forth one paper in men, untrained men, showing that those who gain more ribosomes were the higher responders after 12 weeks of resistance training. So beyond our paper, there's other papers out there. There was a recent series of papers from, somewhere in Europe, Hammerstrom et al., and they showed the same thing. But there's a lot of literature on that, right? So that was hypothesis number 2. Finally, the third hypothesis was higher responders would see a greater increase in satellite cell content versus lower responders. So for the unfamiliar viewers, satellite cells are muscle stem cells. Now, a lot of research has gone into satellite cells and the dynamics of satellite cells with resistance training, or in rodent models, like surgical models of muscle hypertrophy. Where the literature lands right now, and we have a review article I may send to you to put in your show notes, we think that satellite cells are required for hypertrophy as you continue to resistance train. So perhaps not over 6 or 8 weeks, but if you're going to engage in years of resistance training, that continued growth, right, by and large is going to be aided by an increase in the number of satellite cells, and some of those satellite cells will fuse to muscle fibers to add an additional nucleus. So you can have more governors of those muscle cells, and it gets a lot more complicated, but we'll sort of end it there, right? So you need more muscle stem cells in order to hypertrophy was the hypothesis, and we thought that we would see a greater increase in satellite cell number and higher versus lower responders.
Perfect. So heading into it, you kind of on 3 different levels for muscle protein synthesis, ribosomes, as well as satellite cells.
Yep.
You thought you would see some different effects based on higher and lower responders. So with that, is that what you guys saw here? Did you see those differentiated effects, or what did you guys see?
So bout number 1, the muscle protein synthesis response between the higher and lower responders. And I should note, right, we had 34 females, so we were able to clearly delineate 8 higher responders and 8 lower responders. So now we're talking about 8 versus 8 data here. Does that make sense?
Yep.
All right. So that first muscle protein synthesis response to the first bout of training, no difference. So you can mark that off the list, and I can get into why that is later on. Well, and in retrospect, why we think that we didn't see differences there. The second was the change in muscle ribosome content didn't shake. So from pre to post intervention, there actually virtually was no change in either group in terms of muscle ribosome content. The third being satellite cells, we indeed showed that higher responders had more satellite cells after training, whereas the lower responders did not. So at least the satellite cell story sort of, stayed intact, which was great because Marcus Baumann's group has shown that those that show the greatest muscle fiber hypertrophy with 16 weeks of resistance training show the greatest increase in satellite cells. Stu Phillips' lab showed that the first bout of training, if people had a greater satellite cell response, because you can have satellite cell proliferation after a bout of training, those showing that large increase also happen to be higher responders. And so, our paper is agreeing with these areas of the literature. We looked at, so at that point, we were like, okay, we've confirmed the satellite cell story, great. Was there anything else that differentiated higher versus lower responders? And this is also interesting. So you had asked, dietary differences. Well, we looked at self-reported macros, and there were no differences in macros, right? Protein intake per day, carb intake per day, fat intake per day, kcal intake per day. So nothing there. Granted, I get it, man. Self-reported food logs are, questionable at best, but we did our due diligence, and we told the participants to be as truthful as they can, so on and so forth. The second was we asked for menstrual cycle day prior to training, because there's a little bit of literature to suggest that if you train during different phases of the menstrual cycle, you could have more favorable training outcomes. When you compared that data point between high and low responders, no differences. The final was we asked them to self-report sleep in terms of duration per day, and we didn't see any differences there. So that's kind of interesting. Now, granted, both the higher and the lower responders on the sleep front, they were obtaining, I would say, around 9 hours self-reported. So that isn't to say that, well, what if we kick everyone down to 6 or 5 hours, we wouldn't see a differential response. That's certainly true, and I think we need to have more human data on that front. I've talked to researchers in the muscle field, some involved with circadian rhythms, like Kieran Esser is a really big name in that space, and sleep disruptions can not only alter sort of that central circadian rhythm, that originates from the brain, but each tissue also has its own set of circadian related genes. And so if you get sleep deprived, are we offsetting the expression of those genes? Could that trickle over into metabolic genes and genes important for muscle protein synthesis and so on? That all needs to be fleshed out more. What we're saying is that, in our study, both groups got 9 hours of sleep. So that is what it is. But this is sort of differentiator number 2. The higher responders started training with significantly more lean tissue mass. As a matter of fact, they had 7 pounds of more muscle, according to the DEXA, than the lower responders. So this is kind of the notion of, okay, if you come into the gym and you're a bit of a mesomorph, or maybe sort of pseudo-endomorph, right, that you can gain more muscle when engaging in resistance training compared to somebody who may be more of an ectomorph.
Interesting. Very interesting. And on your note of kind of circadian rhythms and stuff, I actually have Dr. Emily Manoogian from the Salk Institute coming on my podcast here in just a couple of weeks. So that should definitely be an interesting conversation. And my master's degree was actually in like chrononutrition and circadian biology and stuff like that. So definitely an interesting topic for me. So that will be a really cool discussion. But to kind of head back to your results here, it sounds like really the main 2 differences between the higher and low responders were the satellite cells, as well as their kind of baseline lean body mass heading into the study, which, the lean body mass one definitely makes sense to me, as I know just anecdotally with my experience with clients and stuff, the ones that kind of come to me that are already like relatively muscled tend to continue responding well to training.
Sure.
Whereas others that maybe they have a really hard time putting on mass, they also tend not to respond quite as well to the intervention there. So is there anything else regarding your results that you kind of wanted to cover here before maybe we get to how this kind of conflicts? We talked about a little bit of how it conflicts with some other research, but anything else that you wanted to discuss regarding the results?
Yeah, so here's what I'll say about that pre-training muscle mass difference, right? So in this cohort of females, the high responders had 7 pounds more muscle versus the low responders. That's great. And that actually agrees with a 1994 study that used a little bit less sophisticated methods. I think it was skin folds, but they showed that, these mesomorphs that didn't have prior training experience but were able to gain more muscle mass through weightlifting versus ectomorphs. We've done these studies in men participants that have come through the lab, and we've observed the opposite, which is to say that I think now 2 data sets would indicate from our men that men who started training with less muscle mass and less muscle size, according to some of that mid-thigh imaging, were the ones that had gained more muscle. Now, that's come under heavy fire because everyone says, oh, that means it's just regression to the mean. So if you test 100 participants and let's say 10 of the lowest on those scales were to train, they're going to sort of regress up. People that maybe came in with, a high amount of muscle mass, maybe they regress down a little bit, and that's where you see that high B low responder. So I thought, well, okay, if certainly regression to the mean explains our past two data sets, we would see it here. And as a matter of fact, we didn't, right? We saw the opposite, which was, so are there gender differences there? That is to say that a young college-age male that is a bit of an ectomorph, sort of ecto-mesomorph, if that person engaged, are they more responsive? To me, it sort of makes sense because I teach this in classes, say, look, hypertrophy happens in phases, as you know, Ryan, right? You have a rapid growth, sort of linear, I don't want to say exponential, but then it certainly tapers off. And then as you approach kind of that natural potential months to years, it certainly tapers and plateaus.
Diminishing returns for sure.
Yeah, for sure. So I said, look, if somebody's kind of that ecto-mesomorph, right, they have a high way to their natural potential. And that's how I used to explain that. But here we're seeing the opposite effect, which I find quite intriguing. So as to why that is from a mechanistic perspective, I can't provide many solid answers, but I think it's certainly exciting and pursuable in the future.
Yeah, absolutely. And one thought I had is maybe if training status would affect that at all, as maybe if they came in with more muscle and they were already well trained, then I would maybe expect them already to maybe be on that kind of diminishing returns curve of muscle growth, to whereas like in this study, if they were relatively untrained, then maybe baseline muscle mass is actually maybe a better predictor of how they would respond, considering like, okay, they haven't really gone down that diminishing returns curve yet. They're still relatively untrained, but they have more muscle. So maybe they have a higher like genetic propensity for muscle growth. Any thoughts surrounding that?
So we actually, this is in men, but our lab collaborated with Stu's lab, and we did a genome-wide association light approach. And that is to say, have you done 23andMe or any of that stuff, Ancestry?
I personally have not.
Okay.
No.
All right. So, what happens there, you spit into a tube, you send off the tube, they isolate the cells in your spit, they get the DNA, and then they subject it to what's called a DNA microarray. There they look for over hundreds of thousands of polymorphisms. So if you have certain versions of different genes across the genome. So we had 110 men between our lab and Stu's lab, and we had muscle. And instead of getting spit, we isolated DNA from muscle because DNA is DNA, right? It's not going to, that genetic code's change from cell to cell to cell. And we submitted that to like a 23andMe type service and looked at hundreds of thousands of genes and the versions of those genes. We really didn't find any genes that were able to predict the response to resistance training, any versions of the gene. So one example would be alpha-actinin 3, right? You can have one of two versions. If you have what's called the RR genotype, that means that your version from mom and your version from dad produce a full-length protein in the muscle cells. A lot of elite powerlifters have this RR genotype. If you have the XX genotype, not many elite powerlifters or Olympic lifters have that. What that basically means is that your muscle makes a short form of that protein that ends up being degraded. And people hypothesize that may diminish the ability to produce force or power, things like that. That's one example of the hundreds of thousands of genes that we looked at across this array, and we didn't see anything. Now, granted, the study that we did with Stu Phillips and these participants in this gene testing, it was only 12 weeks of resistance training. The other caveat I tell people is, look, 23andMe is great. You can get hundreds of thousands of genes and versions of genes worth of data, but we really need deep sequencing, which is, I'm sure you've kept up with it. Now we're able to do some deep sequencing both at the DNA level and RNA level, and we need to revisit and see if we can find novel versions of genes with this deep sequencing to give us more insight there. So I think, genetics, yes, absolutely. There's heritability studies showing that you can have, a heritable contribution to strength or body mass or body size. Those studies are out there. The problem is, where we are right now with the technology, just using DNA microarrays, it's not providing a lot of insight, right? So I think we need to apply new technologies as they come out in order to better answer that genetics question. That was long-winded. So I think your question was, is there a genetic component to this? We didn't test that in these participants, but the answer is we've tested it in men participants, and based on the current technologies to do that, I don't think it provides good resolution, right? That's to be determined.
Gotcha.
Yeah.
Cool. Well, we are kind of coming up on time here, but maybe as just like a finisher here, are there any kind of like practical takeaways regarding some of your research on high and low responders, just kind of in this totality? Like if someone finds that they're not responding very well to training, does this mean that they're just a low responder and they're kind of screwed? Or like what are maybe some practical takeaways here? Yeah,
no, that's a great question. So, and we get this all the time. This is great. You're providing more insight into the markers that exist between high and low responders. Awesome. Another good pub, yada, yada. But at the end of the day, how we change, can we take the low responders and can we make them high responders? So the literature needs to pivot more towards that question, including our own lab. I know Marcus Baumann, they were sort of tinkering with protocols to do so. I'll add one quick note there. There was a Brazilian group, I believe, that did a very nice job with like a 6-month training study where they looked at some sort of surrogate for muscle mass gains and showed that at 3 months, at the 3-month time point, if you were a low responder, if you kept training, so from 3 to 6 months, you stayed a low responder, right? So it's not like progressive overload. It's hard to overcome. Let's face it. So what can low responders do? Well, I think one thing is creatine. I don't get paid by creatine companies. With creatine, monohydrate supplementation, the literature is pretty clear that not only does it help, put on muscle mass, it's debatable because there's fluid shift involved. I get that. But creatine can also kickstart satellite cell activity. So if being a low responder is largely due to satellite cells not proliferating, then perhaps a low responder can take creatine and sort of make that mechanism a little bit more operative. The other thing that hasn't been tinkered with a lot, I think there was a research group, I want to say in Europe, that did this, but taking low responders and then upping the training frequency to see if they could tolerate that more and if that helped them get out of that low response arena. And the answer is, I'm almost positive, I'm going to have to look that up, but I'm almost positive that low responders do benefit a little bit more when they kick up their training frequency. So long as they can tolerate it and they're at a volume that's not, pushing the limits on recovery, then that may be something else to try. And then the last thing, I think doing all this in conjunction, right? Creatine, more volume. But the last thing is to consume more protein, which would, if you just consume more protein and don't change any other aspect of the diet, that is going to give you more calories, more energy, because muscle building is an energetic process, right? You talk about muscle protein synthesis. What people fail to realize is that involves turnover of protein. So you have to break down old proteins, that requires ATP, which comes from, the oxidation of food substrate in the diet. And then every peptide bond, when you're making proteins, requires anywhere between 2 and 4 ATP. And so certainly upping the protein intake, you're giving the muscle substrate for protein synthesis, you're providing more calories. The protein that you don't use, the muscle does a pretty good job of using those carbon skeletons to produce ATP. So at the end of the day, I think if you consume more protein, if you are a hard gainer, aim for a gram of protein per pound per day, right? I'm 220 pounds, so I would aim for 220 grams of protein over 4 meals, right? You're talking about 50 to 60 grams of protein. Consume 5, if not more, grams per day of creatine. You can go up to 10. Darren Candow has a lot of good data in this area. It's not harmful. A lot of research has shown that. And then again, with those two support systems, if you will, you can up the training volume or frequency And instead of working out 2 times per day, you can try to push the envelope a little bit and go 3 times per day. But I think that's where you certainly start if you're a low responder.
Perfect. I think those are great recommendations. And I would definitely agree with the point on just eating enough total calories in general, as a lot of, I would say, people that would classify themselves as hard gainers, they have an issue of eating enough food, and they tend to just be kind of like a high calorie burn kind of phenotype, in which they eat more food and then they just end up burning more food. And they're like also the type of person that, they can't really sit still in a chair. They're always like fidgeting around and stuff. So it's like, okay, we need to just pile in the calories as much as possible. So I think with that, Dr. Roberts, this was definitely an awesome conversation for me selfishly. I really enjoyed it, and I'm sure the audience did as well. So thank you so much for coming on. If people want to stay up to date with the research you're doing, is there anywhere that they can kind of go to make sure that they're staying up to date with that?
Yeah, I tweet. Anytime we put a paper out there, I'll tweet it. So the handle is at Dr. Mike Roberts on Twitter. I have an Instagram with the same handle. I get tagged, but I don't do a lot on Instagram. But yeah, and then Google Scholar, right? So I have a Google Scholar account. And if you were to search, you have to Google Scholar, and then that'll take you to Google Scholar. And then if you search Michael D. Roberts, Auburn, it'll pop up. So, those are the 3. The 2, because I don't do Instagram, but yeah, the 2 ways to do that. Yeah.
Perfect. Well, thank you so much for coming on today.
I appreciate your time, Ryan. Thank you for having me.